Hello, everyone, and welcome to our live MaxCyte-sponsored webinar, Overview of Flexible, Clinically Adaptable Non-Viral Approaches to CAR TCR Methodologies. My name is Leslie Eschinger, and I am the Director of Marketing for MaxCyte. I will be your moderator for today's engaging session. I would like to remind everyone that this event is interactive, so we encourage you to participate by submitting questions at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If at any time you have trouble seeing or hearing the presentation, please click on the Support tab found at the top right of the presentation window. Our speaker for today's session will be Rama Shivakumar, a Senior Research Scientist and Customer Application Specialist at MaxCyte. For Rama's complete biography, please visit the Biography tab at the top of your screen. During today's event, Rama will highlight powerful case studies that demonstrate the successful use of MaxCyte's clinically validated scalable electroporation system in a variety of applications. She will discuss next-generation CAR approaches, compare detailed case studies of both autologous and allogeneic therapeutic solutions, and will examine the advantages of a scalable non-viral approach throughout the development and manufacturing process. Without further delay, let me turn this session over to Ms. Rama Shivakumar.
Thank you so much for the kind introduction, Leslie. Hello, everyone. In this webinar, we will discuss an overview of a flexible, clinically adaptable, non-viral approach to CAR TCR methodologies. The talk is organized in the following segments. We will first go over who we are as a company and how our technology has provided an enabling solution to our customers' research and clinical needs. To illustrate this, we have included 4 specific case studies. In the first case study, we will go over the development of a mesothelin-specific mRNA CAR for both resting and activated T-cell therapies. In the second case study, we will illustrate how this technology was used to engineer NK cells for CAR therapies. We will then talk about the manufacture of Transposon CAR-T cells. And in our fourth case study, we will go over ways to improve the efficacy of CAR TCR therapies with gene editing. We will then round off the talk with a summary. Let us first go over who we are and how we are driving the next generation of cell-based medicines. MaxCyte is a global cell therapy and life sciences company that was founded in 1999. We are headquartered in Gaithersburg, Maryland. MaxCyte's powerful, scalable flow electroporation technology is currently being used to engineer a variety of cell types to enable research, clinical, and commercial applications. Once the transfection process is optimized at the research stage, the high-efficiency reproducible performance can be translated to a clinical or commercial scale rapidly without the need for re-optimization. The same unifying technology can therefore be used to take a product from concept to clinic. MaxCyte's electroporation platform is branded as Expert. The Expert platform is designed to deliver high transfection efficiencies and viabilities to give you a potent cell therapy product that is efficacious and reproducible, which can then enable the rapid clinical translation of your research to a therapy. The Expert is a family of 3 instruments, the ATX, the STX, and the GTX. All these instruments are sleek with a touchscreen and enhanced software user interface occupying very little space on the lab bench. The ATX is research-focused used for small and medium-scale static transfections. The STX is a scalable flow electroporation device that can be used for transient protein production and also in the production of vaccines and other biologics. The GTX is the clinically validated scalable flow electroporation device which has 21 CFR compliance, has a cGMP compliance, and also an ISO certification. We offer many consumables that are compatible with the expert devices. These are called processing assemblies, and one can use them to transfect a range of 75,000 cells to over 20 billion cells. The transfection volumes range from as low as 15 microliters of cells in the smallest chamber up to 100 mils of cells in our large flow electroporation chamber. With these instruments and processing assemblies, we have enabled a number of partners like KSQ, Sangamo, UPenn, CRISPR Therapeutics, Allogene, and Kite, to name a few, to scale very quickly from R&D to patient treatment. We have over 120 clinical licenses, we are engaged in over 90 clinical programs, and we have 11 commercial licenses. In the first case study, we will talk about the development of a mesothelin-specific mRNA CAR in resting and activated T cells. We will show work that was done in Dr. Carl June's lab at UPenn, and we will also show data that was generated from MaxCyte in collaboration with Johns Hopkins University. The goals illustrated in this case study were to determine the therapeutic potential, safety, and feasibility of mRNA CARs directed against mesothelin in activated T cells, and to develop a commercial product with a streamlined cGMP compliance strategy for manufacturing PBMCs transfected with mesothelin-specific mRNA CAR. To determine the on-target off-tumor toxicity of this meso-mRNA CAR, a clinical trial was designed where a cohort of patients were infused repeatedly with the mRNA CAR-T product, and the anti-tumoral clinical response was monitored over time. To develop a commercial product from MaxCyte called as CARmRNA, we simplified the complexity and cost of manufacturing of CAR-T cells, where the anti-human meso-CAR mRNA was directly transfected into PBMCs without prior activation or expansion. In the preclinical study that is discussed, the CARmRNA product was first characterized in vitro and then followed by activity in xenograft models. Patients meeting the eligibility criteria underwent a large volume apheresis to isolate T cells. The T cells were activated by the addition of anti-CD3 and anti-CD28 beads and cultured for 10 to 12 days in a cell culture medium supplemented with human serum. The expanded T cells were then electroporated with mRNA encoding an anti-mesothelin SS1 scFE CAR using the MaxCyte GT system. The CAR-T meso cells were then cryopreserved in an infusible cryoprotectant supplemented medium. A dosing regimen was determined where repeated anti-meso CAR-mRNA doses were first given for several weeks intravenously before switching to intratumoral infusions. Several assays were performed to determine the efficacy of CAR-T infusions. In vivo trafficking. Prior to the commencement of the intratumoral injections, trafficking of CAR-T meso cells to the tumor tissues was evaluated by qPCR analysis. Samples from blood, ascites fluid, and tumor biopsies were collected at several time points. The qPCR results revealed that the CAR-T meso cells had trafficked to the tumor sites when infused intravenously. The CT scan. A CT scan done after the commencement of intratumoral injections showed a distinct reduction in tumor burden. Epitope spreading. When a tumor disintegrates, a number of new epitopes are expressed on the surface. These are then subject to attack by other members of the immune system like B cells, NK cells, and macrophages. The massive immune response results in tumor elimination. To demonstrate this, protein lysates from meso-expressing cell lines were transferred onto a nitrocellulose membrane and blotted with the sera from the patients. The appearance of new bands on the Western blot was consistent with epitope spreading. The results of the preclinical and clinical studies from Dr. Carl June's lab proved that the mRNA CAR approach was safe and feasible with no off-target effects in patients. The process was then further optimized at MaxCyte to reduce timelines by using resting T cells as a viable option for the meso-mRNA CAR therapy. This led to our own commercial product called CARmRNA, where PBMCs are isolated from patients, electroporated with a meso-mRNA CAR, and then frozen in appropriate media until infusion into patients. The CARmRNA CAR construct is a second-generation CAR. It is a CD3 zeta 41BB CAR directed against human mesothelin. The CARmRNA cells induce target-specific killing of mesothelin-expressing ovarian tumor cells like ID8 cells in vitro. The ID8 target cells grew in monolayers when co-cultured with control cells. However, when cultured in the presence of CARmRH meso cells, the ID8 cells were lysed and the only remaining cells were the CARmRH meso cells with small aggregates, which was consistent with activated T cell morphology. To evaluate the activity of CARmRH meso cells in vivo, mice were challenged with ID8 meso tumor cells stably expressing luciferase. On day 4 after inoculation, the CARmRNA meso cells were thawed and injected into mice intraperitoneally. The mice were imaged once a week. Mice injected with 1E7 CARmRH meso cells showed similar tumor signals in the abdomen, which indicated that the treatment had no effect at such a low cell dose. In contrast, mice injected with 1E8 CARmRH meso cells showed significantly lower tumor signals in the abdomen at all time points. A single IP injection of CARmRH meso cells effectively controlled ovarian tumor growth in vivo, but this treatment was short-lived. To overcome the limitation of transient expression of CAR on T cells, the study tested whether repeated IP injection of CARmRH meso cells is more effective in ovarian tumor growth control. The results showed that 6 weekly intraperitoneal injections of 5E7 CARmRH meso cells was more effective in controlling tumor growth than 3 weekly IP injections of 5E7 CARmRH meso cells. This is now an open, ongoing clinical trial. In the second case study, we will illustrate how this technology was used to engineer NK cells for CAR therapies against acute lymphocytic leukemia or ALL and acute myeloid leukemia or AML. The data presented in this case study comes from 2 sources, from Dr. Dario Campana's work at National University of Singapore and Onc Therapeutics in Ireland. The ALL or acute lymphocytic leukemia model presented in this case study is the work done by Dr. Dario Campana's group at the National University of Singapore. Dr. Campana wanted the option of modifying large numbers of NK cells with the anti-CD19 CAR-mRNA in a very short time, because then the infusion of the CAR-NK product into patients could happen in a few hours after collection with minimum ex vivo manipulation and no extra cell culture. The data presented for the AML, or the acute myeloid leukemia model, was generated by Onc Therapeutics in Ireland. To develop a clinically viable CAR-NK product for AML targeting CD38, it was essential to knock out the inherent CD38 in NK cells. This prevents NK cell fratricide. This was then followed by the electroporation of an affinity-optimized anti-CD38 CAR-mRNA. The goal of Onc Therapeutics is to finally make a rapidly scalable, consistent batch manufacture of anti-CD38 CAR-NK product at a low cost that can be rapidly translated into the clinic. The figure on the left is the design of the CAR molecule that Dr. Campana's group tested in this study. The design represents a second-generation CAR molecule with an anti-CD19 specificity. The signaling capacity is provided by the CD3 zeta and 41BB moieties. In a set of 9 experiments, Dr. Campana's group directly compared the transfection efficiency of the anti-CD19 BBZ CAR in primary and expanded NK cells from the same donors. The bar graph in the middle shows the percentage of NK cells expressing the anti-CD19 BBZ mRNA as determined by flow cytometry 24 hours post electroporation. The figure on the right shows the flow cytometric density plots that illustrate the expression of the anti-CD19 BBZ mRNA CAR in 3 out of the 9 donors. Overall, the percentage of cells expressing the CAR was consistently higher in ex vivo expanded NK cells. The median cell viability after electroporation was 90% for the expanded cells and 91% for the primary NK cells. The data for viabilities is not shown. While the receptor expression by retroviral transduction required 2 rounds of transduction on 2 consecutive days, the mRNA electroporation was accomplished in 1 hour and resulted in high CAR expression. The efficiency of CAR expression following retroviral transduction was around 60%, whereas the efficiency of CAR expression following mRNA transfection was around 87%. MaxCyte electroporation was rapid, simple, and Dr. Campana's group was able to overcome viral transduction manufacturing limitations by using the MaxCyte GT system. In the graph depicted on the left, those cells expressing the anti-CD19 BBZ CAR-mRNA exerted a much higher cytotoxicity against a CD19 positive B-cell lineage cell line than the mock transfected cells. The expanded NK cells expressing the CD19 CAR-mRNA were more cytotoxic than the primary NK cells expressing the CAR-mRNA. The figure on the right shows the flow cytometric density plots from 2 different experiments, where the expanded NK cells from 2 different donors were electroporated in the absence or presence of the anti-CD19 BBZ mRNA. The cells were co-cultured in the presence of a CD19 positive cell line in a 1:1 ratio. The results indicate that the NK cells expressing the anti-CD19 BBZ mRNA produced a 2-fold higher level of interferon gamma. In a step towards the clinic, the scalability of the MaxCyte GT platform was assessed, followed by in vivo adoptive transfer to assess anti-tumor activity. mRNA electroporation using large-scale electroporation resulted in 82% efficiency, similar to efficiencies seen in the previous small-scale experiments. Mice were injected with an ALL tumor line, followed 2 days later by the injection of engineered or unengineered NK cells. Those mice receiving the NK cells expressing the anti-CD19 CAR-mRNA displayed a 10-fold lower tumor burden when compared to the mice receiving the control NK cells. Despite the transient nature of mRNA expression, the engineered NK cells exhibited impressive in vivo anti-tumor activity. In the AML study, or the acute myeloid leukemia study, the main approach of onc therapeutics was to genetically modify NK cells to express the affinity optimized anti-CD38 CAR that can effectively eliminate CD38 high expressing AML blasts. It was, however, important to first knock out the CD38 on NK cells using CRISPR-Cas9. This was done on day 5 following expansion. The CD38 knockout was then assessed by flow cytometric analysis on day 30. Following this, the affinity optimized anti-CD38 CAR-mRNA was introduced into the CD38 knocked out NK cells via electroporation. The in vitro functionality of the affinity optimized anti-CD38 CAR-mRNA expressing CD38 knockout NK cells was then assessed in an in vitro assay. The dot plots on the left show a 90% knockout of CD38 post electroporation. The dot plots on the right show a 42% expression of the anti-CD38 CAR-mRNA 20 hours post electroporation in the CD38 knocked out primary donor derived expanded NK cells. 100 micrograms per mil of CAR-mRNA was electroporated in this study using the MaxCyte GT system. The dot plots represent results of the cytotoxicity assays with mock or CAR-mRNA electroporated CD38 knocked out NK cells co-cultured with AML CD38 expressing cell lines like THP1 and KG1A target cells. The CAR expressing CD38 knocked out NK cells show enhanced cytotoxicity. The data presented here are from Dr. Shigeki Yagyu's lab. Dr. Shigeki Yagyu is a professor at the Department of Pediatrics at the Kyoto Prefectural University of Medicine in Kyoto, Japan. Dr. Yagyu wanted to develop a non-viral CAR-T methodology using the Piggybac Transposon system for the treatment of pediatric rhabdomyosarcoma in Japan. In Japan, one cannot use viral vectors like lentiviruses or gamma retroviruses in clinical trials. Sendai viruses are used on occasion, but this too has several drawbacks. Dr. Yagyu was looking to develop a highly versatile clinical manufacturing protocol with reduced costs and timelines to develop a cheaper, safer, functionally better transposon CAR directed against the Ephrin B receptor. This case study outlines the generation of the anti-Ephrin B receptor CAR-T cells and also an anti-CD19 CAR-T product in PBMCs and their in vitro cytotoxic potential. As a preliminary experiment, conditions for DNA loading was optimized in resting PBMCs. A GFP plasmid was used as a DNA control. Increasing the DNA concentration resulted in higher transfection efficiencies as demonstrated by higher percent GFP in the FACS plots. Increasing the DNA concentration resulted in lower viabilities as seen in the 7-AAD staining showing the effects of DNA toxicity. Once DNA loading into PBMCs was optimized, the co-transfection of piggyback transposon and transposase was addressed in an experiment. A total of 15 micrograms, a 1-to-1 ratio of piggyback transposon and piggyback transposase was electroporated into PBMCs at varying cell densities ranging from 1E8 cells per mil, 4E8 cells per mil, and 8E8 cells per mil. FACS analysis was done at 48 hours to determine viabilities and also CAR-positive cells. The CAR-positive cells increased with increasing cell densities. The results were best at 88 cells per mil with the number of CAR-positive cells at 52% and high viabilities at 86%. Here, the MaxCyte process with high cell density electroporations allowed for conservation of resources. 40 million PBMCs were transfected using the MaxCyte optimized protocol with the piggyback transposon and transposase. The cells were then expanded on a layer of UV irradiated feeder cells along with a cytokine cocktail containing IL-7 and IL-15. At the end of the 14-day expansion process, 3E8 efferent-B CAR-T cells were retrieved. They were also able to retrieve 4E8 CD19 CAR-T cells as well using the same protocol. After the 14-day expansion protocol, the FACS analysis showed that the percentage of the efferent-B CAR-T cells were around 67% and the percentage of CD19 CAR-T cells were 82%. These cells also conformed to the desired naive memory T cell phenotype, and they also showed a non-exhaustive phenotype with no expression of the PD-1 checkpoint inhibitor. The potency and durability of the piggyback-engineered efferent-B CAR-T cells were assessed using an in vitro co-culture with the target efferent-B expressing rhabdomyosarcoma cell line RH30. Efferent-B CAR-T cells were plated at a 1-to-1 or a 2-to-1 ratio with 10,000 RH30 cells and incubated for 3 days. An additional 10,000 RH30 cells were added to the culture in the second and third round, and the number of live tumor cells were determined each time 3 days later. The RH30 cells were rapidly killed during a second and third round challenge, demonstrating the sustained activity of the efferent-B CAR-T cells. In our fourth case study, we will go over ways in which the MaxCyte technology can be used in gene editing applications to improve the efficacy of CAR and TCR therapies, and also how this technology enables the making of an allogeneic CAR-T product. Recent advances in gene editing technology allow for the manufacture of CAR-T cell therapies starting from healthy donor leukapheresis material in which the quantity and quality of the T cells can be pre-selected. To decrease the number of alloreactive T cells and prevent graft versus host disease, endogenous TCR, alpha beta, and beta 2 microtubulin genes are knocked out using CRISPR-Cas9, TALENs, or other gene editing tools. The MaxCyte GT system has been shown to knock out the tract locus with greater than 95% efficiency in both Jurkat cells as well as activated T cells. The Cas9 and guide RNAs can be co-delivered as RNA or RNP to get a high degree of knockout efficiency without compromising the viabilities of the target cells. The gene editing efficiencies are carefully optimized with a linear correlation between the total energy delivered and the gene editing efficiency. Researchers at WashU in St. Louis were able to achieve over 95% multiplex gene editing efficiencies using the MaxCyte platform. The conditions were first optimized to maximize the GFP mRNA transfection efficiencies in activated T cells before designing a highly efficient multiplex RNP approach to knock out the TRAC and CD7 loci to up to 95%. Very pleased with these results, the research group at WashU went on to test whether these gene editing efficiencies scaled up seamlessly when a larger volume of cells were electroporated in a bigger processing assembly. The compelling data here shows that the process is effortless and the high efficiency gene editing is consistent and reproducible when you scale up from the smaller OC100 processing assembly to a larger R1000 processing assembly. In a complex experiment, researchers at the Seattle Children's Hospital used the MaxCyte platform to engineer activated T cells using a mRNA AAV co-delivery method. Cas9 and mutant adenoviral helper proteins were delivered as mRNA, followed by the AAV delivery of multiplex guides for the simultaneous knockout of TRAC and a checkpoint inhibitor, TIM3. Mutant adenoviral helper proteins were mainly used to maintain a cellular DNA repair environment that is conducive to gene editing. 80% of TIM3 and 65% of TRAC were knocked out simultaneously in this study. Genome editing, when combined with TCR immunotherapy, has the potential to increase the potency and safety of the engineered T cells. The phase 1 clinical trial illustrated here used the MaxCyte GT platform to electroporate patient T cells with CRISPR-Cas9 RNPs containing 3 single guide RNAs targeting the TRAC, TRBC, and PD1 loci. The edited T cells were then transduced with a lentivirus expressing a TCR specific for the New York ESO1 tumor antigen. The engineered T cell product was then infused into the patient. The engraftment and persistence of the edited T cells in the patients were monitored over time. Autologous T cells from 2 myeloma patients and 1 sarcoma patient were obtained by apheresis, elutriated, washed, and enriched for T lymphocytes. Electroporation of the RNP complexes containing equimolar mixtures of Cas9 with single guide RNAs for TRAC, TRBC, and PD1 were performed as per MaxCyte's standard operating procedures. The RNP complexes were mixed with the desired concentration of T cells and loaded carefully into a processing assembly and then electroporated using an optimized protocol on the MaxCyte GT instrument. The cells were then cultured in the appropriate media with IL-7 and IL-15 at 30 degrees centigrade for a time. Following the gene editing step, the cells were then transferred to a 37 degrees centigrade incubator, activated with CD3, CD28 beads, and then transduced with a lentiviral vector encoding the TCR specific for New York ESO1 antigen. The cells were subsequently expanded in a wave bioreactor with IL-7 and IL-15 until harvest day. The final genetically engineered T cell product was termed as NICE, which referred to the New York ESO1 TCR transduced XXX gene-edited T cells. At the end of the 8 to 12 day expansion protocol, over 1E10 cells were obtained from each patient. The digital PCR approach was used to determine the gene editing efficiencies. The results varied depending on the single guide RNA used. The gene editing efficiency for the track locus was 45%. The gene editing efficiency for the TRB locus was 15%, and the gene editing efficiency for the PD1 locus was 20%. At the ASGCT meeting this year, Dr. Carl June affirmed that the reagents used for gene editing have come a long way from the first generation reagents that were used in this study. They are now getting over 90% gene editing efficiencies for the 3 loci as they embark on expanding this trial to include a larger cohort of patients. The high fidelity on-target editing for TRAC and PD1 was well over 99%. It was a little lower for the TRB locus, but then the gene editing efficiency was lower for this locus as well. When the NICE cells were co-cultured with the target cells expressing the New York ESO1 tumor antigen in an in vitro cytotoxicity assay, the NICE cells demonstrated potent antigen-specific cytotoxicity over a wide range of effector-to-target ratios. In fact, the NICE cells showed even greater cytotoxicity than the control cells that expressed the transgenic TCR but were electroporated without the CRISPR reagents. In another assay to assess the polyfunctional profile, the NICE cells that were activated with the CD3-CD28 beads secreted very high levels of interferon gamma in response to the target cell. The patient outcomes after the infusion of NICE cells were encouraging. The frequencies of the gene-edited and TCR-engineered cells remained quite stable between day 10 and 4 months post-infusion. There was also an increase in the expression of genes associated with the central memory phenotype. These data are not shown here. Samples were collected from patients between 3 and 9 months post-infusion and expanded in the presence of New York ESO1 antigen. Antigen-specific cytotoxicity was assessed by chromium release assays and was observed in all patients. The CT scan from one patient also showed a 50% regression of a large abdominal mass. These case studies illustrate the diverse applications of MaxCyte's technology in the CAR TCR field. The non-viral approach discussed here is robust, safe, and cost-effective. The mRNA CAR therapies in resting and activated T cells and also in NK cells are efficacious with no serious side effects. It is possible to generate potent piggyback transposon CARs that can be translated rapidly to the clinic. Gene editing can not only be used to generate allogeneic cell therapy products, but also to improve the efficacy of an already existing cell therapy product. At MaxCyte, we have a partnership approach to our science. We have a globally distributed, goal-oriented field applications and sales team. We have a very experienced in-house R&D team. We also have a skilled quality and regulatory affairs team who handle our FDA master file and quality documentation. MaxCyte's expert platform is more than just a technology. It is a solution. Thank you for listening and for your patience. I can now take your questions.
All right. Thank you, Rama, so much for your informative presentation. I'd like to use the remaining portion of the session to now ask Rama any questions our viewers might have. So if you have a question you'd like to ask, please just do so now by using the Ask a Question box located at the far left of your screen. All right, let's get started. So Rama, does MaxCyte have experience with other transposon technologies? And if so, what is the technology and what is the performance related to the technology?
Hi, Leslie. Can you hear me? Can everybody hear me?
Yes, Rama.
Okay, great. So yes, the answer is yes. MaxCyte is working with a lot of companies with their proprietary transposon technologies, and also with other PIs, specifically working with Sleeping Beauty-based CAR products and other piggyback transposons or piggyback transposon technologies. So this technology is used in both activated T cells as well as in CD4 and CD8 positive T cells. We have seen from our data from the customers that we do get superior transfection efficiencies when compared to lentiviral transduction of transposons. So I would say like we have efficiencies that are consistent and reproducible, and some of the groups have reported well over 70% efficiency in transfecting transposons.
Okay, next question. How does mRNA-based gene delivery systems work when it's transient? It may not give a sustained efficacy against tumor or relapse. So what is the argument behind this approach?
That's a really good question because I presented so many case studies on mRNA transfection. So the argument is you would think of an mRNA CAR approach as more like a hit-and-run approach, like you would see with bispecific antibodies and other, antibody therapies. But specifically, how the advantages of doing the mRNA therapy is you will have to do repeated infusions of the mRNA CAR product, as we saw in our case studies. And the advantage of that is, as you know, CAR therapies and TCR therapies to a lesser extent, but CAR therapies typically have a lot of serious side effects and cytokine release syndrome and such that are evident when in the prolonged expression of CAR given using viral vector methodologies. So mRNA therapies can control that. So since they are given as repeated injections, one can actually stop the injection when the patients succumb to some kind of cytokine release syndrome. And as evidenced in some of our case studies, we have seen pretty efficacious and robust response that is very target specific and not off-tumor toxicities as seen in the first clinical trial with epitope spreading. So that was really successful.
Okay, great. Rama, you had mentioned the cryopreservation as part of the workflow during the mesothelin CAR case study. What are the effects of cryopreservation on the mRNA CAR modified T cells?
So MaxCyte has a number of cell therapy approaches, not just with CAR TCR, but with other disease models as well, where we use mRNA to deliver the therapeutic product. So we have optimized the culture conditions and electroporation parameters for these delivery of these mRNAs into resting or activated T cells or NK cells. And then we find that when in the course of optimizations, we find that when these cells are thawed, they actually don't lose their CAR potency and efficacy. And so that's how and the viability is also retained. So we promote this kind of therapy using our instruments.
Okay, next question. Does electroporation need to be optimized at small scale using the ATX system before scaling up to the GTX system?
Well, that is a good question. The answer is no, because you can. So the GTX system can be used for electroporating or optimizing conditions by using cells at even 50,000 cells or 75,000 cells all the way to 50 billion cells. So you don't really have to use a research grade instrument like an ATX, to optimize prior to moving to a GTX, you can stick with the same instrument and go from your research scale application all the way to clinical or commercial scale.
Okay, next question. Can you explain how mRNA CAR electroporation compares to retroviral transduction in ex vivo expanded NK cells?
So in the case study that I showed with Dr. Campana's group, he actually did a head-to-head comparison with a gamma retroviral transduction of a CD19 BBC CAR delivered as either a gamma retrovirus or as an mRNA. And in that study, he showed that the CAR expression following retroviral transduction was about 60%, whereas with the mRNA CAR, he consistently got about 87%. And since NK cells tend to be short-lived, he decided to go with that approach as a viable option for his clinical trial.
Okay, great. Another question that came through about construct size. So is there a limit that can be electroporated effectively into cells regarding the construct size?
Okay, is this question in relation to CAR TCR particularly or just in general? So if it is in relation to CAR TCR, the case study that I showed with the Transposon CAR, those constructs are 6 kb roughly each. We do get efficient transfection efficiencies up to 8 or 9 kb of CAR DNA. But then for protein production and other applications, we have electroporated much larger sizes. And for viral vector production, sometimes the size is also very large. So, yeah.
All right, that's helpful. Another question has come through about cytokine release syndrome and its effect on CAR T cell therapy. Can you speak to the cytokine release syndrome effect, Rama?
Yes. So cytokine release syndrome is a common side effect that's found in CAR therapies. It's to a lesser extent in TCR therapies, but basically, when a CAR therapy is really effective, you get, massive, in the course of tumor elimination and recruitment of all the immune cells, you get a release of a number of cytokines, IL-6 particularly, that causes high fevers in patients and is commonly called as the cytokine release syndrome. And that's like a commonly seen side effect in most virally induced CAR therapies.
Okay,
usually
great.
Effective CAR. Yeah.
Okay. Next question. Does MaxCyte's electroporation process affect the cell surface receptor expression on NK cells?
Okay, so that's a good question. So the MaxCyte electroporation process is a fairly inert process. So according to the published data, the people that have used this instrument for not just for NK CAR expression, but for other, ADCC-related studies done in NK cells, they've basically found that there was no effect after electroporation on any of the cell surface receptor expressions like NKG2D or CD16 that was evaluated by FACS analysis post-electroporation.
Okay. Next questions regarding viability. So a customer typed in, what is the viability of the cells 12 and 24 hours post-EP?
So this is in relation to mRNA transfections or DNA transfections?
It doesn't specify. Can you answer both? Yeah.
Okay, yeah. So mRNA transfections is, well, as we all know, in immune cells, mRNA transfection does not affect viability at all, and it's usually well in the 90s, either 6 hours post-transfection or 24 hours post-transfection. DNA, immune cells are pretty resistant to taking up DNA as such, So DNA does affect viability, but it also depends on the concentration of DNA used. So there's a linear correlation between the amount of DNA and viability. So as you increase the amount of DNA, there's DNA-related toxicity. So let's say, for example, 100 micrograms per mil of DNA, you would not see that, which is what we see in Dr. Yagyu's work, which had over 86% viability post-DNA transfection, and that was two DNAs, the transposon and transposase, that were co-transfected. I don't know if that helps.
It does, thank you. Next question. Can the MaxCyte flow electroporation technology be integrated with commonly used culture systems to create a fully closed process?
Yes, so that is a really good question, and the answer is yes. MaxCyte, any of those instruments, the ATX, well, the ATX is usually a research-grade instrument. So the flow electroporation technologies that basically require higher cell manipulations can be sterile welded with other devices like the G-Rex devices that's commonly used for growing T cells, or wave bioreactors, which is done downstream of the electroporation process, or it even is bio-welded quite well with the Miltenyi system. So yes, the answer is it can be sterile welded and can fit into the workflow of a customer pretty easily.
Okay. Next question is about the electroporation parameters. So a customer typed in, can these electroporation parameters be optimized or modified in the expert platforms?
Yes, so this is correct. We do. So the way we work with our customers is typically we provide as much guidance as possible to the cell therapy customers and give them a list of optimized protocols depending on the cell therapy that they are engaged in. But we also provide a list of optimization protocols that the cell therapy customer can use independently of us to try to optimize their own conditions. So yes.
Okay. We have another question that came in. What is about the cytokine release syndrome again? So what is the chance of cytokine release syndrome effect using mRNA CAR-T therapy?
Okay, so that's a good question. So the cytokine release syndrome is sort of proportional to how effective the CAR-T therapy is. So even if you're using an mRNA CAR-T therapy and it is really effective, then you might get cytokine syndrome. But the advantage to mRNA CAR therapies is that can be controlled, which in a viral vector methodology, it leaves your hands once you have infused the viral vector CAR that's constitutively expressed in the patient. Whereas in an mRNA therapy, that can be controlled by the number of infusions that you can give a patient. So if the patient there is visible cytokine release syndrome effects, then the mRNA therapies, the infusions can be stopped. So it's a more controlled environment.
Okay. Next question that came in, does MaxCyte have a cGMP compliant electroporation buffer?
Yes. So we have just one buffer that comes with our system, and that one buffer works with almost all the cell types, except for, I think, with algal cells, where we had to use a different kind of buffer. But basically, the MaxCyte electroporation buffer is cGMP compliant. It's produced by HiClone for us, and they do it with the cGMP, all the regulations that go with the cGMP compliance.
Okay. And then I think this probably might be the last question, unless we get any more after this one. But are there any clinical trials or products in the market using your non-viral approach?
Yes. So as I showed in the talk, we ourselves are engaged in a in a clinical, MaxCyte ourselves engaged in a CARmRNA clinical trial that's ongoing. And we do have a number of customers that are using our technology in a variety of ways in the clinic, whether it's in transposon therapies. They are either well on their way into getting into a clinic, they are working on their pre-IND filing, or they're in their non-human primate studies and will very soon get into clinical trials. Or we are also, as I mentioned in the last case study, a number of our customers are using us in allogeneic approaches that are already in clinic right now. So we are upstream of the CAR process, or downstream where we used to delete the track B2M to create an allogeneic product. So the answer is yes, and there are other CAR therapies that are non-viral therapies that are also well on their way to being in the clinic or already in the clinic.
Okay, great. So it doesn't look like we have any more questions coming in. So with that, I'll go ahead and actually wrap up the session. So I wanted to remind all of our audience members that the recorded version of today's talk will be made available soon after this live session. So you will be notified via email when it becomes available. And also, any questions that were unable to be answered today via the actual live session, the answers will be provided to you using the email address you provided at the time of registration, as well any that come through during the on-demand period. So before we go, I did want to take some time to thank Rama for her very detailed presentation. A thank you to the customers and industry partners for allowing us to share your important work with the larger cell and gene therapy community. And I also want to thank LabRoots and their supportive webinar team for organizing and promoting our event. Lastly, I would like to thank you, our audience participants, for joining us today and for your interesting questions. We look forward to having you in attendance at our next MaxCyte LabRoots sponsored session on September 17th, featuring our scalable manufacturing and nanovesicle delivering solution using CRISPR-Cas9 RNPs. So this will conclude our session for today. Until next time, goodbye.
Bye.